Hello, I'm Dr. Nesmani, and this is the second of the series of concept bridges in physiology. Please visit the site youtube.com/acpgmet. Please also have a link in facebook.com/acpgmet. This is the address, please note down. So let us continue with the cell physiology, cell signaling. In the last session we were talking about the cell membrane. Now we will know how the cells communicate with each other. There are three types of cell signaling: autocrine cell signaling, paracrine cell signaling, and endocrine signaling. Autocrine, paracrine, and endocrine signaling. Autocrine signaling involves a secreted substance acting on the same cell that produced it. The substance is produced and the response is also in the same signal. Paracrine signaling means a substance diffusing from the signaling cell that produced it to nearby target cell to elicit a response. Here the cell produced and the response cells are adjacent to each other. For example, the gastrointestinal regulatory peptide somatostatin is produced by the D cells in the stomach, diffuses to the gastric acid cells, and decreases the secretion of the stomach. Endocrine signaling involves a substance secreted by endocrine cells that is transported in the blood to distant target cells to elicit a response. For example, adrenocorticotropic hormone, which is released from the anterior pituitary into the blood, stimulates the release of cortisol from the adrenal gland. Cell signaling events. A signaling cell produces a signaling molecule termed a ligand or a primary messenger, which binds to receptor associated with a target cell. Ligand binding results in conformational change and activation of the receptor. The activated receptor elicits a response in the target cell either directly or indirectly through the production of a secondary signal termed a second messenger. A target cell response includes alterations in cellular metabolism and alteration in gene transcription. Second messenger examples include cyclic AMP, DAG, that is diacylglycerol, and IP3. Hormone binding to a G protein results in activation of phospholipase C, which catalyzes phosphatidyl inositol 4,5-diphosphate to form IP3 and DAG. Types of receptor classes. Intracellular receptors located in the cytoplasm or nucleus of target cells are bound by lipophilic ligands which diffuse through the membrane of the target cell. Ligand binding alters the receptor's conformation, exposing the receptor's DNA binding domain. Receptors bind specific gene promoter elements and activate transcription of specific genes that results in the synthesis of specific proteins. An example is an estrogen receptor in uterine smooth muscle cells. There are four types of cell surface receptors. Nicotinic cholinergic receptors are linked to ligand-gated ion channels that are selectively permeable to specific anions or cations. Example, nicotinic AChR on muscle cells. Nicotinic acetylcholine receptors on muscle cells. The figure shows the example of cell surface receptor. There is a hormone and receptor. Membrane is also shown. Adenine cyclase, ATP to PP1, cyclic AMP is produced, intercellular enzyme, and there is a biological effect. The second type, catalytic receptors, are transmembrane proteins that have an intrinsic enzymatic activity like serine or tyrosine kinase. Then other receptors linked to proteins with enzymatic activity. These receptors do not have catalytic activity themselves. An example is cytokine receptor signaling through cytoplasmic tyrosine kinase like JAK TYK STAT system. The fourth one is the G protein linked receptors. They have an extracellular ligand binding domain, an intracellular domain that binds G protein. After ligand binding, the receptors interact. G protein linked receptors, they have an extracellular ligand binding domain. That is the fourth one. G protein linked receptors, they have an extracellular ligand binding domain, an intracellular domain that binds G proteins. After ligand binding, the receptors interact with the G proteins. G proteins are heterodimeric, consisting of alpha, beta, gamma, and subunits that dissociate. Figure on the right side, maybe please note that for some time all G protein coupled receptor proteins span the membrane 7 times. 7 clusters of amino acids in the plasma membrane represents hydrophobic portion of the protein's alpha helix. Exterior domains are called E1, E2, E3, E4. Cytoplasmic loops are called C1, C2, C3, C4. Amino acid residues in the third cytoplasmic loop nearest the C terminal interact with the G protein. The G proteins alpha subunits bound to GTP interact with and activate specific membrane bound enzymes, resulting in the production of second messenger that elicit response in the target cell. An example is adenylyl cyclase system. Clinical correlation: cholera toxin alters G protein so that guanosine triphosphate GTPase is unable to hydrolyze GTP, resulting in increased production of cyclic AMP. Elevated cyclic AMP in intestinal epithelial cell result in massive gut secretion of water and electrolyte, resulting in severe diarrhea and dehydration. Pseudohypoparathyroidism. In pseudohypoparathyroidism results from a defective G protein and causes decreased cyclic AMP. Patient exhibits symptoms of hypoparathyroidism with a normal or slightly elevated parathyroid hormone level. Whooping cough and pertussis toxin blocks the activity of G1, allowing adenylyl cyclase to stay active and increase cyclic AMP. Now we will talk about membrane potential. The membrane potential is the difference in electrical potential or voltage between the inside and outside membrane surface under resting condition. Cells have an excess of negative charges at the inside surface of the cell membrane and exhibit a negative membrane potential at rest. Because potassium concentration inside the cell is higher than the outside concentration, potassium moves out of the cell leaving excess negative charges on the inside of the cell membrane. The sodium-potassium pump acts as a second factor to generate negative charges on the inner membrane surface by pumping 3 sodium out and only 2 potassium in. Potassium efflux is primarily responsible for the resting membrane potential. The equilibrium potential is the membrane potential that exists if the cell membrane becomes selectively permeable for an ion, causing the distribution of the ion across the membrane to be at equilibrium. Then Nernst equation, N-E-R-N-S-T. Nernst equation describes the relationship between the concentration gradient of an ion and its equilibrium potential. Thus, an equilibrium potential is predicted by the Nernst equation, which is given there. Here the E is the equilibrium potential in volt, R is the gas constant, T is the absolute temperature, F is the Faraday's constant, Z is the valence of the ion, 1 for sodium, 2 for calcium. In is the logarithm to the base C, Co is the outside concentration of the positively charged ion, Ci is the inside concentration of the positively charged ion. In nerve cells, the resting membrane potential ranges from minus 80 millivolt to minus 90 millivolt, which is near the potassium equilibrium potential. Therefore, nerve cell membrane are selectively permeable to potassium. The Nernst equation predicts that the equilibrium potential for potassium will be negative because K0 is less than Ki. Ko is less than Ki. It also predicts that the equilibrium potential for Na plus will be positive because Na0 is greater than Nai. Because the membrane is most permeable to potassium and chloride, the actual membrane potential in most cells is around minus 70 millivolt. Resting membrane potential is the potential difference across the cell membrane in millivolt. The resting membrane potential is established by different permeabilities or conductances of the permeable ions. For example, the resting membrane potential of the nerve cell is more permeable to potassium than to sodium. Changes in ion conductance alters currents, which change the membrane potential. Hyperpolarization is an increase in membrane potential in which the inside of the cell becomes more negative. Depolarization is a decrease in membrane potential in which inside of the cell becomes more positive. Hyperpolarization, inside cell becomes more negative. Depolarization, inside of the cell becomes more positive, or negativity decreases. Both are the same. An action potential is a rapid, large decrease in membrane potential, that is depolarization. The figure shows the action potentials. Membrane potential in millivolt on the vertical axis, time on the horizontal axis, and the recording shows the rest, depolarization, overshooting, repolarization, threshold, and the rest. Action potentials usually occur because of the increase in the conductance of sodium, calcium, and potassium ions. Threshold is the membrane potential that induces an increase in sodium conductance to produce an action potential. Depolarization produces an opening of the sodium channel through the fast opening of the activation gate, slow closing of the inactivation gate. Closure of the inactivation gate results in closure of the sodium channel, decrease the sodium conductance. Slow opening of the potassium channel increases potassium conductance higher than sodium conductance, resulting in repolarization of the membrane potential. Thus, repolarization is the return of the membrane potential to its original value due to an outward potassium movement. The refractory period is the period during which the cell is resistant to a second action potential. During this relative refractory period, only some of the inactivated sodium channels are reset and potassium channels are still open. Thus, another action potential can be elicited if the stimulus is large enough. Propagation of the action potential requires a system that regenerates the action potential along the axon. Conduction velocity is increased by increased fiber size, myelination, and is dependent on the magnitude of the depolarizing current. Myelinated nerves exhibit saltatory conduction, in which the action potential skips from one node to another node where the voltage-gated sodium channels congregate. That's a node of Ranvier. Depolarization block occurs when depolarization stimulus occurs slowly so that sodium channels may inactivate before enough sodium channel opening occur. Thus, even though membrane potential exceeds the threshold, no action potential is produced. Organophosphate poisoning occurs by depolarization block of neuromuscular junctions, thereby inhibiting acetylcholinesterase from breaking apart acetylcholine molecules. Organophosphorus, there is depolarization block of neuromuscular junction. Acetylcholinesterase is inhibited. Acetylcholine molecules are not broken down. It accumulates. Structure of skeletal muscle. Skeletal muscle is organized into a progressively smaller anatomical unit. Muscle fibers are surrounded by a plasma membrane, more commonly called sarcolemma. Muscle fibers are composed of a bundle of fibrous structures called myofibrils, and each myofibril is a linear arrangement of repeating structures called sarcomeres. Sarcomeres are a fundamental contractile unit of skeletal muscle. They are characterized by their highly ordered appearance under a polarizing light microscope. Sarcomere structure is shown here. The A band contains the thick filaments. The I band contains the thin filaments, which are attached to and extend from the Z line. The Z line maintains the regular spacing of the thin filaments within sarcomere. The space between terminations of thin filaments is called the H zone. The denser area within the H zone is called the M line. Thick filaments in the A band are composed primarily of the protein myosin. Each myosin molecule is composed of 6 monomers, 2 protein strands intertwined in a helical arrangement termed heavy chain, 4 small globular protein called myosin light chain, and there are 2 essential light chains and 2 myosin regulatory light chains. Each heavy chain is associated with a globular head. The 2 globular head of myosin heavy elements can hydrolyze ATP to ADP and inorganic phosphate, and also have the intrinsic ability to interact with actin. The rod-like region or tail stabilizes the protein and tends to self-aggregate spontaneously, thereby forming the thick filament. Treatment with the proteolytic enzyme trypsin splits myosin into 2 components, heavy muromycin light myromycin. Another proteolytic enzyme, papain, cleaves heavy myromycin into globular protein S1, rod-like protein S2. The site sensitive to proteolytic digestion are regions that allow flexing of the molecule, also called hinge region. Thin filaments are composed of three primary proteins: actin, tropomyosin, troponin. Actin can exist in two states: globular G-actin, filamentous F-actin. G-actin polymerizes to form F-actin. Each G-actin monomer contains binding site for myosin, tropomyosin, and troponin I. Basic structure of thin filament consists of two strands of intertwined F-actin in a double helical arrangement. Tropomyosin is an elongated protein that lies within the two grooves formed by double-stranded F-actin. Each thin filament contains 40 to 16 tropomyosin molecule. Troponin is a complex of three separate proteins. Troponin T binds the other two troponin subunits to tropomyosin. Troponin C binds calcium, the crucial regulatory step in muscle contraction. Troponin I is responsible for the inhibitory conformation of tropomyosin-troponin complex observed in absence of calcium. The thin filament structure is shown here. The actin filament, tropomyosin, troponin, calcium binding site, active site, actin. Then myosin filament, tail, heavy myromycin, light myromycin, head, the actin binding site, and ATP binding site. If you go to the YouTube and type the muscle contraction animation videos, you can get animation pictures showing the contraction of muscles, cells at this protein level. Tubules, a tubular network, are located at the junctions of A bands and I bands and contain a protein called the dihydropyridine receptor. The sarcoplasmic reticulum SR is the site of calcium storage near the transverse tubules, T tubules. It contains a calcium release channel known as the ryanodine receptor. The transverse tubules, T tubules, contain a calcium release channel known as the ryanodine receptor. We talked about this in the previous video on malignant hyperthermia. Several steps are involved in the mechanisms of muscle contraction. Action potentials in muscle membrane cause depolarization of the T tubules, which opens calcium release channel in the sarcoplasmic reticulum, increases the intracellular calcium. Calcium releases the troponin tropomyosin inhibitory influences so that the active sites on each G-actin monomer are uncovered. Myosin globular heads that produce from the thick filament bind with the G-actin active sites, thus forming cross bridges. Intra-molecular forces stored energy within the myosin molecule allow myosin to flex in the so-called hinge region. These areas are the two proteolytic enzyme-sensitive regions in the myosin molecule. The action of flexing of the myosin molecule causes the globular head, still attached to actin, to tilt towards the center of the sarcomere. This movement, called the power stroke, creates tension that results from shortening of individual sarcomere. Immediately after the tilt, the cross bridge is broken. Globular heads snap back to the upright position. At this point, a new cross bridge can be formed if the ATP and calcium are available in the vicinity of thick and thin filaments. In the absence of calcium, cross bridge formation is not possible. Relaxation occurs when calcium uptake into the SR lowers intracellular calcium. Biochemical events that occur during muscle contraction cycle involve an active complex and Riger complex. Myosin with ATP bound to it, that is ATP. Myosin ATP complex has a low affinity for G-active protein site. When calcium binds to troponin and tropomyosin rotates out of the way so that the active site on G-actin are uncovered. Myosin ATP is simultaneously hydrolyzed to myosin ADP, which has a high affinity for G-actin active sites. Consequently, an active complex or crossbridge is formed between actin and myosin ADP. ADP is released from myosin, globular head tilt towards the center of the sarcomere, producing tension. At this stage, Riger complex is formed between actin and myosin. ATP then binds to myosin. Myosin ATP complex breaks the crossbridge. Globular head snap back to upright position. The cycle is ready to start again in presence of calcium. Skeletal muscle enters a state of prolonged stiffness called rigor mortis after death. Rigor mortis occurs because with death, muscle cells are no longer able to synthesize ATP. In the absence of ATP, cross bridges between myosin and actin are unable to dissociate. After 15 to 25 hours, proteolytic enzymes released from lysosome begin to break down actin and myosin. The practical aspects of filament interactions involve the relationship between muscle length and tension. In an isometric contraction, muscle length is held constant during the development of force. An example would be an individual pushing against an immovable object such as the wall of a house. In isotonic contraction, the muscle shortens while exerting a constant force. An example would be an individual lifting a glass of water to his mouth. The tension that a stimulated muscle develops when it contracts isometrically, that is total tension, and the passive tension exerted by the unstimulated muscle vary with the length of the muscle fiber. The difference between the two values is the tension produced by the contractile force, the active tension, that is shown in the figure. Amount of active tension developed with a contraction decreases from its maximum as the muscle is either shortened or lengthened prior to the contractile stimulus. The length-tension relationship is the relationship between the length of the muscle and the amount of active or passive tension in the muscle. Active tension refers to the tension generated by the contractile forces when the muscle is stimulated, whereas passive tension refers to the elastic forces acting on the muscle when the muscle is stretched. Total tension on the muscle is the sum of the active and passive tension. So on your left side, you can see that just muscle length and stimulate measure the tension, and on our right side the vertical axis shows muscle tension, horizontal axis shows the sarcomere length. There are three curves: total tension, active tension and passive tension. Active tension developed is proportional to the number of crossbridges formed. Tension is reduced when the sarcomere is shortened to a point where thin filaments overlap, prevent one another from forming crossbridges with myosin. Thus, isometric tension produced depends on the degree of overlap of the thick and thin filament, which dictates number of crossbridges that can be formed. The force-velocity relationship refers to the relationship between the load or weight placed on a muscle and the velocity at which that muscle contracts while lifting the load. Velocity is the distance an object moves per unit time. Load can be thought of as a weight that the muscle is attempting to move via an isotonic contraction. For example, when a weightlifter tries to lift a series of progressively heavier weight. A muscle can contract most rapidly with no load. As load increases, however, the velocity at which the muscle lifts the weight decreases. When the weight equals the maximum amount of force that the muscle can generate, velocity becomes zero. In this case, the contraction becomes isometric. For example, the muscle contracts but does not shorten. The functional unit of a muscle is called motor unit. Motor unit consists of one motor neuron, axon, all the muscles innervated by that motor neuron. In adult, each muscle fiber is innervated by a single motor axon. In general, motor units in small muscles that react to stimulation rapidly and subserve functions that require fine control have a low number of muscle fibers. An example is laryngeal muscle, in which motor unit has approximately 2 to 3 muscle fibers per motor neuron. Motor units in large muscles that subserve functions is not requiring fine motor control tend to have a large number of muscle fibers. An example is gastrocnemius, in which a motor unit contains approximately 500 muscle fibers per unit of motor neuron. Because all the muscle cells in a motor unit contract together, fundamental unit of contraction of a whole muscle is the contraction produced by a motor unit. Increased tension development in the skeletal muscle is attained by wave summation, like increasing stimulus frequency of a single motor neuron, summation or recruitment of motor units. Besides increasing tension development, recruitment allows a movement to be continuous and smooth because different motor units fire asynchronously. That is, while one unit is contracting, another might be at rest. A contraction can be single, brief contraction or maintained contraction due to continuous excitation of muscle fibers. Single contractile event or a twitch is initiated by a single action potential from a motor neuron reaching the neuromuscular junction. If a second stimulus is applied before the muscle fibers in the motor unit have relaxed, second contractile event builds on the first. It can be said that the two contractions summate. This summation of contraction occurs when stimulation frequencies reach about 10 per second. As the frequency of stimulation is increased, developed force continues to sum until a maximum developed force is reached. At this point, the individual contraction relaxation cycles fuse to produce a single smooth curve called a tetanus. Tetanus occurred in skeletal muscle because the refractory period is short relative to contraction time. Refractory period is the time during which the tissue does not respond to a second stimulus. The graph shows the contractile force and the stimulus and the tetanus produced. Recordings of contractile force during twitch contractions and the tetanic contraction of skeletal muscle are shown. A twitch contraction is a single brief muscle contraction that occurs in response to a single threshold stimulus. Tetanic contraction or tetanus is a constant contraction of skeletal muscle due to continuous excitation of muscle fibers. Hope that the session was useful to you. Next session will be on neuromuscular and synaptic transmission. Goodbye from Dr. N.S. Mani from ACPGMAT. Please visit the channel youtube.com/ACPGMAT. And there is the email address acpgmat@gmail.com. Please visit the channel every day. Thank you.